My name is Sarah McDonald, and I'm an assistant professor at the Virginia Tech Carilion Research Institute with an appointment in the Department of Medicine at the Virginia Tech Carilion School of Medicine and also at the Virginia Maryland Regional College of Veterinary Medicine at Virginia Tech. So I've always been fascinated with viruses because they're so basic, but they're so complex and they have such a huge impact on human health and disease. So at their simplest, viruses are just vehicles, little capsules, either envelopes or proteins that have within them strands of nucleic acid. And even though they're so simple, they're able to usurp our entire cellular machinery. So it's almost like if you have a factory that makes one certain kind of widget, let's say it makes, for example, radios, and the virus is able to go in that factory and now make that factory make telephones instead of radios. So it can literally take over our cellular machinery to use to its benefit. And we study a virus called Rotavirus, and the reason why we study Rotavirus is really twofold. One is that it remains a really important pediatric pathogen. It's responsible for several 100,000 childhood deaths worldwide. It causes diarrhea in infants and children. It's estimated about 40% of diarrheal deaths worldwide in children are attributed to rotavirus infections. And really the reason why these kids are dying is based on the fact that they don't have access to medical care that could save their lives. But also, it's unique in that it replicates its nucleic acid genome in tandem with assembly. So it actually needs to coordinate these tasks during the infectious cycle. And so we're really interested in understanding, how does the virus do this? What are the interactions that are occurring? And also, what is the structure of these replicase assembly intermediates, if you will, these viral assembly intermediates that are enzymatically active? And can we then use that structure to infer things about function of these different molecules that comprise these assembly intermediates? So that's really the major thrust of that research grant. So securing NIH funding has been instrumental for me to recruit a very talented research team. This is a team that's comprised of postdoctoral fellows, graduate students, medical students. I also have medical residents who work on several aspects of the projects in my lab. And not only is it important for my line of research, but it's also critical for them to get the training and skills and expertise that they need to then go on and make discoveries of their own. So I think most scientists who are in positions like I am, in leadership positions, always hope and wish that their trainees will then go on to make even better and more important discoveries than what they made. And so that's something that's really important to me. And without the NIH funded research, I would not have that pipeline in place. So it's critical. It's exciting in that there might be a discovery that I make that I can't even imagine will have an impact, but certainly the ones that I can imagine are the more we understand about how viruses assemble, the more insightful we can be about targeting different steps of this assembly process to prevent viral disease. So certainly if we can halt the virus from assembling, we can halt the virus from replicating, and we can halt disease progression. But I'm also interested in this understanding of if we can utilize what we know about virus assembly to actually use viruses to our advantage. For example, in gene therapy for delivery vehicles, or if we can actually use them as nanomaterials by taking advantage of their self-assembly properties. So I think that what we find out could be informative on many levels. It's hard for us to predict exactly how the path of discovery is going to lead to the innovation that could save many lives, but I would argue that history suggests that that path has been trod many, many different times in many different ways.